Hello, everyone. My name is Gabriella Petricelli, and I worked with Mary Person for our biology senior research project. We looked at the effects of environmental enrichment on rehabilitated owls in captivity. Our research advisor is Dr. James Kellum.
Stress can be elicited by an unnatural situation, like captivity, and it can manifest itself in a wide manner. Stressful or traumatic circumstances activate the hypothalamic pituitary axis in birds, and the adrenal gland consequently releases cortisol and corticosterone into the bloodstream. One option to measure stress levels in birds is to evaluate the amount of circulating corticosterone, their primary stress hormone, in their system. The assessment of this avian glucocorticoid coincides with the bird's physical response to the stressor. Unfortunately, high amounts of corticosterone can have a harmful impact on a bird's health, fitness, and reproduction if enduring chronic stress levels. These consequences are evident in both captive and wild birds. Environmental enrichment can be used to see if there are successful or unsuccessful forms of sensory stimulation. Successful environmental enrichment is when one bird interacts at least once with a novel item. This sensory stimulation can cause the owl to be more active while hopefully decreasing their stress levels. We started conducting the research footage on October 31st of 2020 and ended on January 10th, 2021. Due to technical difficulties, we do have missing footage from some of the weeks involved in this time frame. The corticosterone assay done to measure the hormone was done during the spring of 2021. This study was conducted at Wildlife Works Incorporated, located in Youngwood, PA. This is a wildlife rehabilitation center that helps animals either get placed back into the wild after injury or gives animals a place to live due to being non-releasable. They also house opossums, squirrels, and other birds. The owls we worked with are non-releasable. Gabby and I were both interested in this area of study because we both have been interested in veterinary medicine, along with wanting to improve animals' qualities of life. We felt that it would be interesting to work with birds of prey due to not having much experience with them before as well. Upon modifying the enclosures of 3 rehabilitated and non-releasable owls, we predicted that the owls would exhibit fewer stress-indicative behaviors and produce reduced levels of corticosterone relative to their degree of tameness. The younger barred owl received the most human interaction, and we therefore hypothesized that he would react the most enthusiastically to enrichment with the least stress. The screech owl was slightly less socialized, so we expected her to show less interest in enrichment than the barred owl and display higher levels of stress. The older barred owl was the least accustomed to life in captivity, and we suspected that he would demonstrate the least dramatic response to an enrichment environment with the highest indication of stress.
Wildlife Works' 3 resident owls varied in age, gender, species, and history. We would take 2 weeks to monitor their routine behavior before introducing enrichment in the form of a swinging perch wrapped with artificial grass, a mirror, and a tub of fallen tree leaves. Over the course of 10 weeks, we collected fecal samples weekly and recorded the owl's movements daily using trail cameras. The fecal samples were used for hormonal analysis using a corticosterone assay, and we reviewed the footage to compare the owl's movements both before and after adding enrichment to their enclosures. First up, we have Shakespeare, a 27-year-old male barred owl. A barred owl's natural lifespan is generally around 18 years, but they can live even longer in captivity without the threat of hunters or food competition. Not much was known about any of the owls' lives prior to coming to the wildlife rescue, but of the 3 in our sample size, Shakespeare's the most wild. He receives the least amount of human interaction and doesn't participate in educational or awareness programs like the other 2 owls.
The second owl we worked with is named Spirit. Spirit is a female screech owl that is approximately 4 years old. The lifespan of a screech owl is between 13 to 14 years old, but this can vary depending on habitat location and captivity status. Spirit is the middle owl when it comes to tameness level. She does have some forms of human interaction while being at the facility, but she is rarely used for any awareness or educational programs. The third owl we worked with is named Artemis. Artemis is a male barred owl that is approximately 4 years old. He is the tamest of the 3 owls. He is frequently used for educational and awareness presentations, or at least he was used for those before the times of COVID-19. I am sure he will get back to those programs soon.
Here is a graphical representation of Shakespeare's activity during our project, comparing both pre- and post-enrichment periods. The behaviors we recorded as we watched videos are listed on the y-axis, and they include walking, ruffling feathers, and flying. As you can see, overall, he spent less time engaged in these actions without the enrichment. Once we added the enrichment in his enclosure, we noted his activity per day increased for almost all types of movement. This doesn't necessarily mean his behavior became repetitive, which could indicate stress, but it does objectively demonstrate that he was more active post-enrichment. Figure 2 takes the data that were on the previous slide and shows only the average number of times Shakespeare performed actions which decreased after the addition of enrichment: hooting, head bobbing, scratching his face with his foot, and preening. We rounded to the hundredths place when calculating daily averages, and even though it appears to be the same, hooting decreased by 0.01 post-enrichment. We wanted to emphasize these 4 actions because they could be considered stress-induced behaviors in captive birds, and seeing them become less frequent once Shakespeare was given things to interact with could suggest a decrease in his stress levels. Here's a sample video recording from the pre-enrichment period. Some things you'll notice Shakespeare doing are hooting, changing the direction he is facing, and shifting his weight. In this video, Shakespeare is clearly interacting with the new mirror on the wall. There are a handful of other instances like this post-enrichment where he faces his reflection before taking off in flight. Here, Shakespeare lands on his new swinging perch, which was tied to the sturdy perch he was already familiar with. We were hoping this would mimic the sway of a tree branch, and as you can see, he finds his balance naturally.
Looking at figure 3, you can see the spirit's activity level with each action increased post-enrichment. The only exception to this is the 180-degree turn decreasing, which is second from the bottom in this figure. This could mean that she was more active due to having more options to choose from, or it could be due to increased stress. Her hormone levels went down, as you will see later, so we hope that it is the first option. You can also see that she stared into the mirror slightly more than she stretched, so this makes the mirror a successful form of enrichment in this case. The box of sand quickly turned into a box of leaves for all of the owls due to there being evidence of them eating it. Although we cannot see it in the footage, the volunteers openly told us that they used the sand and the leaves, so this was also a successful form of enrichment for all of the owls. In this video, you can see the spirit bobbed her head and changed where she was looking, so then she could fly to another area of her enclosure. She then turns around and stares outside the rest of the time. She stared outside a lot during our research, likely looking for movement and listening to sounds from outside. Here, you can see Spirit looking at herself in the mirror. She also flies onto the other perch that was added. Although she did not use the swinging part of this new perch, she did seem to enjoy having another place to sit. Both the perch and the mirror seem to be successful forms of enrichment in this case. Looking at figure 4, you can see that Artemis's activity level with each action decreased post-enrichment for every single action performed. After watching the videos, we feel that this was due to him not liking the swinging perch. In most videos, he did not want to hang out on the new perch and instead walked around on the ground. However, he did interact with the mirror, so this was a successful form of enrichment. In the post-enrichment videos, we were unable to see the box of sand. Again, the box of sand that changed into leaves was also a successful form of enrichment. In this video, you can see that Artemis moves his feet a lot on the perch, while also doing things such as changing where he looks and bobbing his head, preening, and ruffling his feathers. Looking at this video, you can see the new swinging perch. He avoids the perch, but he looks into the mirror at himself. In this video, you can see him bobbing his head, flying across his enclosure, and stretching out his neck. We included this video so that you could see what happened when he flew onto the perch. We feel that this may have been too much movement for him due to being in captivity for a while and not being able to land on swaying branches.
Figure 5 summarizes our corticosterone results. A reminder that stressful environments activate the HPA, which causes the bird's adrenal gland to release corticosterone. We expected to see a decrease in corticosterone levels post-enrichment for all 3 owls relative to their degree of tameness, but as you can see, that's not what happened. We had predicted Artemis, the most socialized owl, to respond to the enrichment with the least stress. Instead, Spirit's corticosterone levels decreased after introducing enrichment, while Artemis's levels barely changed at all. Spirits were also the only data that were tested statistically to be marginally or somewhat significant. We had predicted Shakespeare's corticosterone levels to decrease with enrichment too, although the least since he's the least tame of the owls. However, his corticosterone actually increased post-enrichment, suggesting that he was more stressed than before, at least according to the hormonal analysis. and you can see there the exact values that were obtained from the assay. Unfortunately, there isn't a lot of literature on normal owl corticosterone levels, so we don't have much to compare our owl's control or pre-enrichment values to. In this research project, we intended to quantify stress through behavioral and hormonal analysis. Unfortunately, very little research has been conducted on owl stress response, so it's difficult to say whether we successfully measured stress in our study, and therefore we cannot reject nor retain our hypothesis. Our behavioral results really seem to reflect the variability among the individual owls, who again are different ages, sexes, and species. Because our project did not require us to interact directly with the owls, they were taken care of and handled per usual by Wildlife Works staff and volunteers on a daily basis, which was not a procedure that we controlled. The inconsistency of our results could also be attributed to the age of the fecal sample. The blanket on the bottom of Artemis's cage was cleaned out every morning, which guaranteed a fresh sample. However, since Shakespeare and Spirit each had outdoor enclosures, we would source pellets from the gravel or the tree branches within, which were not changed weekly and meant that we may have collected stool from an older week, which could skew our corticosterone data. Lastly, we faced some pretty significant technical difficulties with the surveillance cameras that we used throughout the project. Most of Artemis's post-enrichment behavior was not captured, and there were a few weeks of missing footage for Shakespeare and Spirit as well, when, despite troubleshooting, the cameras failed to record correctly. So again, our study was pretty inconclusive, but there are a few points in scientific literature which correlate to our findings. The fecal method that we used for our hormonal analysis was nice because it was very non-intrusive, but feces have more than just the corticosterone component, which can make it difficult to isolate just one hormone. And so in 2018, a group of researchers studying fecal metabolites in African gray parrots determined that the most accurate procedure was to dry the droppings and then combine corticosterone extractions with methanol before using a corticosterone EIA kit to obtain concentration values. And we did both of these things to prepare our fecal samples. The researchers also recommended that the stool should be collected at the same time each cycle, since regular release of corticosterone is dependent on the bird's circadian rhythm. However, we were unable to visit Wildlife Works at the same exact time each week. Furthermore, previous research generally shows that compulsive behaviors, such as excessive feather plucking, are a sign of distress and occur due to a lack of decision-making available in the bird's environment. After all, this is an animal that would regularly fly around all day and hunt and explore in the wild, but in captivity, birds spend their waking time, up to 8 to 12 hours, just alternating from perch to perch. The 3 owls in this project did not appear to exhibit any pattern of repetitive behaviors, but Shakespeare was observed to vocalize less after enrichment was added, and Spirit was seen flying around more, which might mean they were less stressed than before. On the other hand, Artemis spent much less time preening post-enrichment, which could indicate increased stress. Each owl, though, was at some point observed interacting with one or more forms of the provided enrichment, so the curiosity was there. If we were to repeat this study, we would place multiple cameras in each of the enclosures to minimize footage loss and maximize visibility from every angle to get more from our behavioral data. It would be beneficial to observe more than 3 participants as well, especially ones so different from one another, but our sample size must depend on the population of captive owls that is accessible to us. A survey that was conducted in 2016 to over 600 wildlife institutions in the U.S. investigated which methods of enrichment were most effective among imprinted or tame and non-imprinted or wild owls. Successful forms of enrichment were considered any that at least one owl interacted with at some point. Meanwhile, types of enrichment that at least one owl ignored or that distressed at least one owl counted as failed. By these standards, reflective objects like mirrors and perch variety were proven to be successful more often than not. This was also observed in our study. The results of this survey indicated that there is in fact a slight correlation between imprinting and engagement with enrichment, but the effectiveness of the enrichment methods addressed in the survey did not significantly differ between imprints and non-imprints. We also did not see as much of a difference as we expected between our owls, who were each at a different degree of tameness. However, survey responses suggested that the younger, non-imprinted owls seemed more interested in enrichment than those older. Therefore, if we were to conduct further research on the subject of owls and stress in captivity, we would propose studying the effects that age and even gender might have on responsiveness to enrichment. Ideally, we take what we learned from this research project to explore how to reduce stress in man-made environments in an effort to improve the quality of life of other wild animals in captivity.
We would specifically like to thank our advisor, Dr. James Kellum, for all of his help during the project. He allowed us to explore our ideas, all while making sure we were not harming the birds or causing too much stress in the process. We would also like to thank the staff at Wildlife Works for allowing us to use their animals and facility. Thank you to those involved in allowing us to obtain the A.J. Palumbo Endowment, which helped us to pay for all of our supplies. Lastly, we would like to thank our families for being so supportive throughout our college career, including during this research. We would not have been able to do this project without the help of all of these individuals. We appreciate you watching, and please leave any questions or comments you have underneath this video. Thank you.